Alright, welcome everyone. Now we're going over part 2 of the cell cycle, which is mitosis. So as a review, the cell cycle starts with the interphase, growth phase, then moves on to mitosis and cytokinesis, and also has a G0 rest phase. So in interphase, the cells are growing, carrying out its normal functions, and has replicated its DNA and all that we discussed previously. In mitosis, which we're going to delve into deeply here, the cell actually is going to undergo cell division, and the cytokinesis is going to actually split into two separate cells. And in G0, as we had mentioned before, it's just doing its normal functions. It's not dividing at all. So mitosis has several distinct steps, 5 specifically, and those are prophase, prometaphase, metaphase, anaphase, and telophase. So you can easily remember this with the acronym PPMAT, with the prophase, prometaphase, metaphase, anaphase, and telophase in that order. So starting off with prophase. So this is the cell that we got at the end of interphase. Everything is it's all duplicated. The organelles are duplicated. They're not shown here. But yeah, basically the cell is now ready to begin dividing, considering it has passed all of the previous checkpoints. So in prophase, the centrosomes are going to move to opposite sides of the cell. So one centrosome is going to move here on the left, one centrosome is going to move here on the right. And then the cohesin proteins, which are holding the twin sister chromatids together, they're going to be broken down like a little bit, but we'll delve into more how that happens later. The nucleus and nucleolus, they're also going to break down, which is key here for the chromatids to actually be separated later on. And the chromatin actually condense into the chromosomes that we all know, that familiar X shape. So prophase in sum, the nuclear memories are going to break down, being that the nucleus and nucleolus. The centromeres are setting up on opposite sides to prepare to release spindle fibers, which we'll go into more depth later on. And the sister chromatin are going to condense into chromosomes. Alright? But how do they condense into chromosomes? So let's look at it more closely here. So here we have two sister chromatids. One chromatid here on the left, which is, as we remember, is one double-stranded DNA. It's very long, it's uncondensed. And we have another one that's also one double-stranded DNA. Remember, they're in a usable form in this period. So when they're long and they're not condensed, they can be used and expressed, alright? That's going to change in prophase. So we see this cohesin holding them together. That's going to break down. But it's going to leave a little bit remaining in the middle to hold them together to form the chromosome later on. Alright? So as we've seen so far, the two uncondensed strands, each of them being a sister chromatid, they're being held together by a little bit of cohesin proteins in the middle. Now more proteins are going to come and attach, and these are called histone proteins. Basically, think of this like beads on a string. So the histone proteins attach like beads on the string, which is for the DNA and the chromatids, to wrap around. So basically, each chromatid is going to like wrap around those proteins in a certain way. And this forms clumps of the uncondensed DNA on the histones, which is now starting to become condensed. So as we can see, these nucleosome stacks are going to come together and like condense everything. So now each of the sister chromatids, they can no longer be expressed because they have been condensed and shortened so much, alright? So and then they start to form together and become that X shape of the chromosome that we all know. And that little bit of cohesion in the middle, that is going to be called the centromere, which is basically holding the whole chromosome together. And on the ends of the centromere are going to form kinetochore proteins, which are basically hooks on the ends of the chromosome. Alright? So as a quick review, I know that was a lot. So the cohesin proteins holding the two sister chromatids when they're very long together are going to break down, but some remain to make up the centromere, which still holds them together like glue. Alright? So after that cohesin breaks down, histone proteins are going to come and basically clump around each of the chromatids' DNA to form nucleosomes. And those nucleosomes are going to stack to shorten the chromatid. So there's like, think about like putting beads on a string. You put all the beads together on the string to form the nucleosomes, and then you like pull the string down and becomes shortened because the beads are getting closer together. That's basically what's happening here. And then kinetochore hooks form on the chromosomes. Alright, so now we got all the chromosomes to condense, meaning they can no longer be used. So by the next phase, prometaphase, the nucleolus and the nucleolus are completely gone. They are not visible anymore. They're not in the picture. Alright. So now this is really where the centromeres are gonna shine. So they are going to release spindle fibers to anchor themselves in placement of the cell, and those spindle fibers specifically are called asters. So we're going to release these little brown aster fibers over here to anchor themselves to the side of the cell and lock in place. They're going to release more microtubule fibers which are called non-kinetochore microtubules. They're basically just released as they are. But then the really vital thing here is the kinetochore microtubules. So in prometaphase, the centromeres actually release the microtubules, which are called kinetochore microtubules, and they latch on to the kinetochore hooks of the chromosomes that we mentioned before. So think of it like fishing. So the centrioles are basically like fishers. They're going out into the sea and they spot a little chromosome that's like a fish there. So they cast their fishing line, which is like the kinetochore spindle fiber, and they latch onto it with the hook that is on that little chromosome. Alright? So basically, in prometaphase, the centrioles/excentromes, they release 3 types of microtubule spindle fibers: asters to anchor, kinetochores to hook, and then non-kinetochores for the cytoskeleton later on. Alright, now becomes the next phase, metaphase. And this phase is easily recognizable under a microscope because all the chromosomes are aligned in the center vertically like this in a random order using the tension of the spindle fibers. And there is a checkpoint here, and that verifies that all the sister chromatids are correctly attached. So like all the hooks are there, all the fishing lines are cast, and they've all caught on. Alright? That's basically what happens in metaphase. And then in anaphase, which is a very short phase, the non-kinetochore microtubules are going to stretch apart the cell as they push into each other. So that is basically stretching out the cell like this and creating lots of tension. On the other hand, the kinetochore microtubules, they're going to shorten, which is going to split apart the chromosomes into their own sister pieces. So think of it like a rubber band. When the rubber bands are pretty long, right? But when you let go and shorten, they pull everything back with them, which creates like a lot of force that snaps the chromosomes apart. And the centromeres and cohesion, therefore, they break apart completely. The glue is no longer there. And this helps maintain the chromosomes have an identical set because remember, sister chromatids are twin sisters. They are the same strands of DNA that has been condensed. So that basically is evenly distributed across the cell. So we can see red here, corresponding red here, blue here, corresponding blue here. It's like a mirror. Put like a mirror in the middle. They're basically the same. Alright? So 2n still equals 4. The diploid number is still the same. So in sum, non-kinetochore microtubules push apart the cell in anaphase, which gives the cell like this oval shape. And the kinetochore microtubules are going to shorten, like kind of retract a bit, and split the chromosomes into 2. Now we get to the final phase of mitosis, telophase. So all the microtubule spindle fibers, they're really going to just break apart and release the chromosomes. And the chromosomes, now they're going to go to the centers of their respective cells. So you can see red ones here, blue ones here, blue ones here. They're going to go and center themselves which allows for the formation of the nucleus and the nucleolus as well. So now you can see like these two cells actually forming and becoming relevant. The cell membrane is going to pinch a little bit also in telophase, which gives it like this recognizable shape here. So in telophase, the chromosomes center in their own cells. Microtubule spindles are breaking down, which allows for the formation of the nucleolus and nucleus, as also the chromosomes are decondensing a bit to become back into chromatids. Then finally, in cytokinesis, as we can see, there are now two cells formed, and each of these cells, if you remember from the previous video, they look exactly the same as they were in G1 in the previous video. So the whole process can start again. All right. So as a review, how does mitosis ensure that each daughter cell receives an identical set of chromosomes? Pause the video and answer. Okay, and we're back. So before mitosis begins, the cell replicates its DNA in S phase, leading us back to that interphase video, creating two identical sister chromatids, like twins. And during these mitosis, they're aligned perfectly in the center. There's a checkpoint that makes sure of that. And then separated by spindle fibers, which gives each new cell the same identical sister chromatids corresponding to the other cell. Alright, thank you for watching. I hope this was helpful, and make sure to subscribe.